one of the students looked at me as he walked by and said, "You are going to freak out," which is usually a good thing to hear before one of these meetings.
Hello and welcome to the Cheeky Science Show, where in this video I had the pleasure to interview David Liu, Professor at Harvard University, the Broad Institute and Howard Hughes Medical Institute. David's lab is using chemistry and evolution to develop next-generation therapeutics. They've developed base editors and prime editors to study and hopefully treat different genetic disorders. From his work, David has co-founded many different biotechnology companies, including Editas Medicine, Pairwise Plants, and Beam Therapeutics. So I hope you enjoyed the following interview. But first, I need to deanimate, not deaminate, as we'll learn about later. What does this do? Perfect. So hi, David. Thank you so much for joining me today, and welcome to the Cheeky Science Show. You are officially the first ever guest, and I'm particularly excited because it was probably the first video that did quite well for me on my channel was all about prime editing, which is a gene editing technique that was developed in your lab, which we'll come on to a bit later. And so it feels like I've kind of gone full circle a bit here. So it's just great to have you on.
Oh, well, I'm really honored to be your first guest, and I'm a big fan of your videos and your channel. And I think you do a great job explaining complicated science to lots of people. So thank you for doing that.
Thank you. So speaking of genome editing, what was your introduction to genome editing? Because I think most people, when they think of it nowadays, they think about CRISPR, but obviously CRISPR wasn't the first genome editing technique that was used. So what was your introduction to genome editing?
Right. So, as is often the case, the route that we follow in science is a combination of our curiosity, but also a lot of serendipity and the network of chance meetings and friends that you happen to have. So in our case, our lab has always, since it started in 1999, specialized in using sort of the components of biological evolution, including in vitro selection, mutation, replication, to address problems in biology and in therapeutic science. At the time, I had a graduate student, Vikram Patanayak, who was working on an in vitro selection system that could identify RNAs with certain biological properties. And while he was sort of thinking about what project he should do next, he happened to attend a Harvard Medical School event. He's a medical school student, was a medical school student, and happened to sit next to Keith Jun, now my good friend, collaborator, colleague, co-founder of a number of companies. And simply by the good fortune that he sat next to Keith Jun, they ended up talking about an area that Keith, was really and continues to be a leader in, namely zinc fingers. And what Vikram became interested in and confirmed with Keith that this project idea was significant was that zinc finger nucleases had already started to enter the clinic. Sangamo was running clinical trials on CCR5 cleaving zinc finger nucleases. Yet, to our knowledge, there was no unbiased, broad characterization of the DNA specificity of whole, intact zinc finger nucleases. What researchers had done as a surrogate before was to take individual zinc finger arrays and do in vitro selections against libraries of DNA to see what they bound. But you could imagine, as I think has proven to be the case for many DNA binding proteins, that the sequence requirements for nuclease cleavage are not exactly the same as the sequence requirements for DNA binding. So this led, to make a long story short, to a collaboration between our lab and Keith's lab. That was our very first gene editing. In fact, it wasn't even called gene editing then. It was called genome engineering. And then it became genome editing, and now it's called gene editing. But our very first project in that field, which was to develop and apply an in vitro selection method that could very broadly characterize the DNA cutting specificity of zinc finger nucleases in this broad, unbiased manner, where we took advantage of the fact that you can make large libraries of circles of different DNA sequences, and when they're cut, they get linearized. You can capture the linear ends. It's actually related to some of the current methods used to characterize the off-target DNA cleavage activities of CRISPR and TALENs and zinc finger nucleases. And indeed, after publishing that first paper on zinc finger nucleases with Keith, we published a second on TALENs and a third on Cas9, which was a collaboration with Jennifer Doudna's lab, our first with her lab. And so our early foray into gene editing was really in trying to achieve a deep understanding of their DNA specificities of these agents and how to improve them. And in some cases, we were able to use the knowledge we gleaned from these unbiased specificity profiling approaches to improve the specificity of these programmable DNA binding domains. So we started, in other words, by simply trying to understand these agents before we ended up engineering and eventually creating our own.
Cool. Yeah, so I was going to say, I think you've highlighted, so when I was first introduced to these zinc finger nucleases, the major, I guess, the big problem with it is trying to identify a way of understanding the interaction of the proteins with DNA and how that specificity is achieved, as you mentioned. As by having that information, you can then, in a way, kind of reprogram those nucleases so that they can recognize other sites of interest. And so that's kind of where, I guess, CRISPR comes in. And instead of using a protein DNA interaction, it uses an RNA DNA interaction, which we understand a lot better in terms of being able to reprogram RNA sequences. And so it gave much more control over the specificity of targeting the editing complex. And so when I first learned about CRISPR, I think I had a kind of wow moment. I definitely remember the moment when I read either the paper I watched Jennifer Doudna talk about it on a TED Talk. I think, I was a bit late to learning about CRISPR technologies. And so I guess, do you remember how it felt when you first learned about CRISPR?
Yeah, it was a kind of aha moment. I think the very first time I started reading CRISPR papers was after hearing about Fanjan's one of Fanjan's talks or papers, where a colleague said, "Hey, you should really check out this area. It seems like it's getting pretty interesting." And I remember having a similar aha moment where we had been working quite a bit with zinc finger nucleases and TALENs by then. I think there's no question that the main barrier to zinc finger adoption as a more broadly used biological tool is that there is not really a simple lookup table you can use to assemble effective zinc finger arrays. You can make reasonable candidates that way, but to get them really good, you need to basically do a zinc finger library in vitro selection and a lot of optimization and to have a lot of know-how. And there are a handful of people in this world who can actually do that. And then, of course, tail arrays, I think, became more popular quite quickly because they were actually more or less de novo designable using the lookup table that various scientists, including Bogdanove, uncovered. So when CRISPR came on the scene and the lookup table was basically Watson-Crick based pairing, I think it did get me very excited that perhaps this was the moment that addressing the genome in this digital programmable way would finally be realized, not just for a company or for a handful of academics with specialized expertise, but maybe for the general life sciences. So it was really exciting. I think nobody back then could have predicted just how democratized it would end up becoming. But it was the start of one of the great revolutions in the life sciences, something that does not come along very frequently. It might come along once a generation or so.
I completely agree. CRISPR has definitely revolutionized, as you say, not only potentially the way we can treat different genetic diseases, but also just as a research tool, with CRISPR screens to investigate and understand how the cells are working. But like with CRISPR-Cas9, the canonical system, in addition to zinc finger nucleases and TALENs, the way that they work is by inducing double-stranded breaks. And so double-stranded breaks are quite deleterious for a cell. It's a major form of damage for a cell. And it's had raised concerns in different papers about inducing the DNA damage response in terms of the p53-mediated response. And so there's concerns over not only basically the safety of having an editing machinery that induces a double-stranded break. And so that's led to finding solutions that edit without having a double-stranded break that might be potentially safer in case there was any off-target editing going on. And so was that why you ended up discovering base editing, or how did base editing come about? Could you explain a bit about that?
Yeah, it's a great question. So as I think you just gave a great concise summary of double-strand breaks and their role in gene editing. It really began with Maria Jason's seminal discovery that a double-strand break can stimulate increased rates of homologous recombination at the cut site. And because the use of the connection between double-stranded breaks, which is cutting a chromosome into two pieces, and gene editing really began with Maria Jason and others discovering the relationship between double-stranded breaks and homologous recombination products, I think there was great hope early on in the gene editing field's history that being able to make targeted double-stranded breaks with these programmable nucleases, whether they're zinc finger nucleases or TALENs or CRISPR-Cas9, would provide a way to do precise gene correction. Unfortunately, we now know that this process of homology-directed repair, or HDR, as it's called, which is the way that a double-stranded break can be turned into a precise edit, is unfortunately not general. And virtually no therapeutically relevant cell types support HDR with an efficiency that would be considered useful at least for therapeutic applications. As a research tool, it is widely used, and in vitro, under a variety of conditions, you can find cells that support HDR. But, perhaps tellingly, I think when I checked last month, so February 2021, somebody asked me, how many CRISPR clinical trials are there right now? And the answer is, so I looked it up, there's 43 of them. How many of them are trying to use a double-stranded break to induce precise gene repair through homology-directed repair? The answer is 0. All of the therapeutic trials using CRISPR right now are using CRISPR to disrupt genes, because the branch of fate that happens once you make a double-stranded cut in a chromosome is predominantly, in most cell types, to lead to gene disruption through indel formation, meaning the cell is desperately trying to get the ends of the broken chromosome back together. And in the process, it will often get the ends together perfectly, but then the nuclease can just chop up the site again, and you go back to the broken chromosome. So every now and then, the act of joining the ends back together makes a mistake, and those mistakes slow down the process of subsequent cutting. So you see an accumulation of those dead end products, collectively called indels, because they have insertions and deletions. It's mostly deletions. It should be called del-ins. It's just more awkward to say, I suppose. And so, to be clear, making indels in a targeted gene can be quite useful, but you have to get kind of lucky if that is your therapeutic strategy. One really important example where scientists, where biology did provide that luck is sickle cell anemia and beta thalassemia, some blood diseases caused by homozygous mutations in the adult hemoglobin gene called beta globin. You can compensate for those mutations, not by fixing directly the beta globin genes. I mean, you could do that in principle. That's just harder to do it with a nuclease for the reasons we just described. But instead, you can use gene disruption to rescue some of those diseases by disrupting a gene that is involved in the silencing of fetal hemoglobin. So, as I'm sure you know, and your audience is probably becoming increasingly familiar with, we're born with redundant sets of hemoglobin genes. We have a fetal hemoglobin set of genes that is responsible for most of our hemoglobin when we are gestating as fetuses. And then around the time of birth, it begins to be silenced, and adult hemoglobin gene expression begins to be turned on instead. So researchers have identified many of the genes involved in silencing that fetal hemoglobin. And if you can disrupt those genes, then you can actually turn back on fetal hemoglobin. And almost everybody who has defective beta globin genes has healthy fetal hemoglobin genes. So it's probably a reasonably strong selection during embryogenesis. So even if you lose the genetic lottery and you inherit or are born with two defective adult beta globin genes, you can still lead a normal life if you have mutations that disrupt silencing of fetal hemoglobin. A very lucky number of people, a small, very lucky number of, well, you could argue they're lucky and unlucky, were born with all of those mutations. They have mutations in their adult hemoglobin that should give them serious blood diseases, but they also have mutations in the genes that are necessary to silence fetal hemoglobin, so they end up being okay. And so a bunch of really smart scientists and companies have exploited this observation to use CRISPR-Cas9 or other double-strand break inducing programmable nucleases to disrupt the genes that silence fetal hemoglobin. So that's an example of how gene disruption can actually benefit a patient. But the reason I said you have to get kind of lucky is, there are a number of small miracles that all had to take place to create this perfect storm of a situation where gene disruption can rescue a genetic disease. Most of the time, in order to benefit a patient, we need to fix a mutation, not simply disrupt it further. The mutation has already disrupted the gene or created a version of the gene that leads to health problems. And in most cases, simply disrupting the gene further isn't expected to benefit patients. Instead, we need to mediate precise gene correction instead of gene disruption. So I think that's sort of a background that, I don't think is any kind of great new insight. It's been known in human genetics for a long time that the nature of most mutations is that they need to be fixed or complemented rather than further disrupted in order to rescue a disease phenotype. But the story about how base editing began then took that background and married it again with the serendipitous prospective postdoc candidates application. This is Alexis Comore, now a professor at UCSD. Alexis was a prospective postdoc, interviewed in my group. Our lab was very impressed with her. I made her an offer. And as I often do with postdocs, even before they arrive, we began to exchange emails about, okay, what projects did you work on? So when they arrive, they can hit the ground running. And so I asked her, what are you interested in doing? And she emailed me a list of sort of proto-project ideas, one of which I thought was kind of interesting. She was proposing the use of puff proteins, which are sort of programmable RNA-binding proteins to mediate changes to RNA. And, I told her, I think this is interesting, but what would potentially be particularly transformative, think I used fairly grandiose language in my email to her, which I've now been asked to dig up several times by journalists. And I think this was back in 2013, so it was a long time ago. 2013, I told her, if you can develop a way to do chemistry directly on a base pair to change one base pair to another base pair in DNA, given the challenges that were beginning to be understood with homology-directed repair and making double-stranded breaks, I think the wording I used was, you would transform the life sciences and quite possibly medicine, or something like that. And so we began to brainstorm how to do this. And she pointed out. So we quickly focused on cytidine deaminases. I think I had proposed that she look at some of the enzymes known to turn C's into U's, and U's, of course, pair like T's. She raised the, I think, very good point that these deaminases seem to work on single-stranded DNA, and of course, most genomic DNA is not single-stranded. And then, as we thought about what different DNA binding proteins to use, I think in a subsequent email, I pointed out that the single-stranded requirement of most deaminases doesn't bother me too much, because Cas9 is known to unwind the DNA into single strands. And, I think at the time that sort of made logical sense, but I didn't fully appreciate how enabling that simple ability of Cas9 to create a single-stranded DNA R loop when it engages the target DNA sequence was. And, as a sort of perfect storm of molecular interactions, Cas9, because it has to pair the guide RNA, Watson-Crick style, with one of the target DNA strands, it has to unwind the DNA double helix so that one strand can pair with the guide RNA, and that leaves the other strand as a sort of free single-stranded DNA bubble. And it's within that single-stranded bubble that base editing occurs on the, quote, non-targeted strand. So the CRISPR biologists named the strand that pairs with the guide RNA as the targeted strand. But for base editing, it's actually the other strand that gets edited first. So that was the basic idea. And we even, early on, talked about how we would probably have to impede the uracil DNA glycosylase enzymes that otherwise are very highly evolved to look for uracil in DNA and cut it out. Because cytosine turns out to be the least stable of the 4 DNA bases. And, your roughly 1 billion cytosines in each of your cells undergo spontaneous deamination at a rate that results in several hundred of them every day becoming uracil. So that would probably be an unacceptable level of mutation were it not for the fact that your cells, and I think just about every kind of cell, has evolved very efficient mechanisms to constantly look for these uracils in DNA and cut them out, and then replace them with Cs. So we identified a small protein inhibitor that would shut down that process, called uracil glycosylase inhibitor, that we could localize to the site of the base edit to try to block uracil glycosylation, at least transiently at that site. And then the final piece came as an insight that Alexis had following her discussions with other group members. So at that time, we had the cytidine deaminase piece, the Cas9 piece, the uracil glycosylase piece, and we were getting okay amounts of base editing, maybe 10 or 15% editing. Thanks to the UGI, it was reasonably clean. But we were trying to get the process to be more efficient, and we speculated that the ceiling of efficiency was probably going to be 50% using that architecture, because if you create a UG mispair, the cell has to figure out whether the U is the correct information or the G is the correct information. And it's going to resolve that disagreement either in your favor, creating a UA base pair, which is what you want, or it'll go back to the starting material to create a CG. And And so we thought, well, maybe we could, and this is really the insight that Alexis brought to my attention, that she had a hunch that if you nicked the G-containing strand, that would cue mismatch repair, which in eukaryotic cells uses nicks to signify newly synthesized and therefore less validated DNA. That the nick would cue mismatch repair to replace the segment containing the G with the newly synthesized DNA segment. And because there was a uracil on the opposite side of the double helix, when it was resynthesizing that new DNA segment, it would put in an A opposite the U. And that would complete the conversion of the information on both strands to what, from what used to be a CG base pair now to a UA or a TA base pair. And so that adding that simple nick, which required just a single mutation put back into our BE2 base editor, resulted in BE3, which was the first, reasonably efficient cytosine base editor. And we showed that it was now able to, I think in our original paper, edit 30 or 40%, sometimes 70%, but clearly showing you could actually exceed 50% editing by biasing that mismatch repair process, which would otherwise, to a first approximation, be a coin toss, to instead favor the repair of both DNA strands the way you wanted. And now, of course, base editors have been improved by many labs, including our own, over many generations. And, we've, in some recent experiments that multiple people in the lab have done at multiple different target sites for multiple genetic diseases, they get close to 100% editing. So it's really come a long way. And, there were, in fact, a new first-year graduate student named Lily in the lab has achieved 100% editing at multiple target sites, including in patient-derived, in patient fibroblasts, primary fibroblasts. So base editing has come a long way. And there were moments early on, even right when we published the paper in 2016, where we wondered, are we ever going to be able to achieve, 80, 90, 100% editing? Even though I think for the vast majority of genetic diseases to benefit patients, you don't need editing that efficient. But the answer actually is yes, you can get close to 100% base editing using the variety of improvements that the community have now advanced from our original work in 2016. And I should also mention that two groups in Japan, Professors Nishida and Kondo, working together, reported in Science several months after our initial base editor paper that Alexis led, reported in Science a similar system where they used a different deaminase, but also with Cas9, also nicking the opposite strand, and reported similar results. So that was also welcome news because it really showed that independent groups were finding the same outcomes, that you could now make for the first time targeted single base pair changes without cutting the DNA double helix, without cutting a chromosome into pieces in a living mammalian cell, in a living human cell. And that really, of course, got us fixated on, okay, let's figure out how to fix as many possible types of mutations that are responsible for genetic disease, and that researchers might want to install in cells to study the properties of genes as possible. But the boundary conditions are you can't make double-stranded breaks.
Well, no, that's really incredible to hear the backstory to how base editing started. And it's incredible also to hear that you're approaching 100% efficiency, which is definitely pretty amazing considering where it started with. And so obviously you've now taken base editing to Beam Therapeutics, which you co-founded, and you're trying to now take the basic science discovery and taking it to being able to treat different genetic diseases. And so out of the different genetic diseases, what do you think base editing will be used for first in terms of getting to patients in the clinic?
Yeah, well, so Beam has disclosed a variety of programs that they plan to bring to the clinic this year or shortly thereafterwards. And based on their announcements, they've disclosed that they are they have developed base editing strategies to treat some blood diseases. They've developed base editing strategies to treat some metabolic diseases as well, like alpha-1 antitrypsin deficiency. And I think they've also disclosed that they have been able to base edit to correct some genetic alleles that are involved in some blindness diseases as well. I always want to make sure that anything I'm sharing is something that they've already shared. So just point people to the Beam Therapeutics website, where they, I think, list the programs that they've publicly disclosed. But I think maybe from a more meta perspective, a decision that I admire that Beam has made is they recognize that the platform was so broad in its potential applicability to diseases that they would not focus on just one organ, one type of disease, one delivery modality, but instead, Beam has, I think, done a great job trying to advance as many programs as possible to cover the broadest possible range of diseases and therefore of patients that could benefit from base editing technology. And so they have really chosen to pursue as many programs that meet some key criteria, like, is there bulletproof biology that relates the correction of the mutation to the correction of the disease? Is there a delivery modality that is promising to deliver the base editors to the cells that are anticipated to lead to disease rescue if corrected? What are the potential base editing strategies that can fix the mutation with the best current base editing technologies known? And, fortunately, I think is a sign of the progress that has led to literally hundreds of base editors now being published, having been published. That third criteria is increasingly proving easier and easier to meet. That with all of the different Cas9 PAM variants, the different Cas proteins that have proven to be compatible with base editors, including a variety of Cas12 variants, that targeting is increasingly not an issue, and even sort of positioning the target nucleotide with respect to the base editing window has also been simplified, not just by the fact that there are now lots of different PAM sequences that you can target, that is, lots of places in the genome where you can park a base editor, but researchers have started to have some success controlling where the base editing window is and how wide or how narrow it is. And so that diversity of literally hundreds of base editors that are available now greatly increases the chance that if you have a mutation you're trying to correct, you can do so with a base editor without too much trouble.
Wow, that's super exciting. Because that's one of obviously the concerns with base editing is you obviously have Cas9 coming into the DNA site, opening up the DNA region, and obviously in theory there could be potential for off-target editing of different sites. So having control over that window is a great advance.
Right, and researchers have shown that you can both mutate the deaminases to control that window, and you can also mutate the Cas9. And in maybe intellectually, at least my favorite solution, you can circularly permute Cas9 proteins, something that Dave Savage has had success with. And so we collaborated with his lab and showed that these circularly permuted Cas9s do indeed change enough the topology of the R loop relative to the deaminase that you can move, in some cases broaden, in other cases actually move over the base editing window. So having a sort of bookshelf with lots and lots of different books that each have slightly different properties has proven to be especially useful for enabling base editing to serve as many purposes as possible at as many target sites as possible, with as many performance characteristics as is needed to enable the research or the therapeutic application of interest.
Wow, no, that's super exciting. And so obviously the base editing has given us a massive toolkit then, but in terms of the actual editing, it's still obviously only a base editing. Even with multiplexing, it's just editing specific bases. And so obviously in terms of precision genome editing, there's still the kind of search for being able to make larger scale genomic changes that we have control over instead of forming indels. And so I think that kind of nicely leads on to prime editing and what prime editing is and what kind of challenges it was trying to solve.
Right. Yeah. So to fill in that story, I think it's a great segue. So the Alexis Comor led cytosine base editor can only make two kinds of DNA changes. It can change C's into T's and G's into A's. And so our first effort to really broaden the applicability of precise genome editing without double-stranded breaks was to develop a different kind of base editor, an adenine base editor, which can change A's into G's and T's into C's. So it actually does the opposite of what a cytosine base editor does. That just because of the nature of human gene mutations, that adenine base editor turns out to be the most useful kind of single base pair corrector, if your goal is to correct mutations that are responsible for pathogenic changes in cells for diseases. So about half of pathogenic disease point mutations are caused by the kind of base pair change that is fixed with an adenine base editor. And so Nicole Godelli, a former postdoc in the lab, now at Beam Therapeutics, developed this adenine base editor that changes AT base pairs to GC base pairs. And there, the project was, I give her a lot of credit because the key enzyme to deaminate adenine in DNA wasn't known. There was no known, and there still is no known naturally occurring reported enzyme that will deaminate adenine into inosine in DNA. And so Nicole agreed to take the brave step of evolving her own enzyme as the starting material for this project, even though in general in our lab we stay away from projects where step one is evolve a starting material, because it sort of sounds like two significant projects, not one. And so after a lot of hard work, she did manage to evolve the first enzyme, to our knowledge, that can deaminate adenine in DNA and convert it into a base that pairs like G. And then we could install that new deaminase that Nicole evolved into a base editing architecture, and then that led to the adenine base editor. And then we've further evolved it, which led to these superactive variants like ABE8E. And that's the variant that we and others are now seeing consistently at some sites in certain kinds of cells, more than 90% editing. But even with the cytosine base editors that Alexis developed and the adenine base editors that Nicole developed, that's only 4 kinds of base-to-base changes. And of course, simple math will tell you, because there's 4 different DNA letters and you can change each letter to 3 other kinds, that means there's 12 total ways you can mutate one base to another base. So the 8 remaining mutations, which are called transversions, we could not make with base editing, at least not efficiently and directly. And of course, there are a large number of insertions and deletions, the vast majority of which are small, fewer than 10 or 20 bases, that are associated with really important genetic diseases like cystic fibrosis and Tay-Sachs disease. So it's been a longstanding goal of the group since we started working with base editors to figure out a way to directly install those other kinds of base-to-base changes and to install deletions or insertions, again, without making double-stranded breaks. And that led to the development of prime editing, which can achieve those goals in mammalian cells. So prime editing really began also with a prospective group member, a prospective postdoc named Andrew Anzalone, who applied to the lab. He's a former MD PhD student at Columbia. He worked with Virginia Cornish, who was a former lab mate of mine when I was a grad student in Peter Schultz's lab. Virginia was a postdoc in the lab. Virginia recommended Andrew strongly, and when he came to the lab at the end of his meetings with various people, I have a one-on-one meeting with him right before he gives his group meeting talk, and I asked him the usual questions. What other labs are you interested in? What kinds of projects do you envision working on? What's the time frame of your desired postdoc, et cetera? And when I asked him, what are you thinking of working on? He pulled out this kind of audacious idea that he had written up into a slide, basically, which proposed that perhaps we could use a reverse transcriptase to directly copy an edited DNA sequence from an extension on a guide RNA into a nicked target DNA site. And, it was a wild idea, and there were all sorts of reasons why it might have failed. It really took a confluence of, what at the time we called small miracles, but now I think just to be more accurate, was just a reflection of the hard work of Andrew and coworkers to overcome. But I was excited enough by the idea that I asked him to do something I normally don't ask prospective postdocs to do, which was, why don't you present that idea at the end of your postdoc seminar? So he gave his group meeting seminar about his PhD work, and at the end, he put in a slide on his idea. And, as you might imagine, the lab found it really interesting, but was also very skeptical about all sorts of various pieces of it, including the fact that when you nick DNA and you put on edited DNA, the cell has to figure out what to do with that edited DNA flap, which is now redundant with the non-edited sequence that's still on the DNA double helix ahead of that nick. And even if that edited DNA flap somehow ends up sitting down on DNA, you now have a heteroduplex where one strand is edited, one strand is non-edited, but the edited strand, the one you care about, was recently nicked. So isn't that going to be preferentially removed by mismatch repair, et cetera? The answer to all those questions actually turns out to be yes, but it still works. So to make a long story short, Andrew started in a test tube where we like to start out, since our group is historically made of chemists and we have strong chemistry ties. So we like to start out in a test tube where we can control and analyze things easier. His idea seemed to be plausible in a test tube. In a test tube, he showed that a Cas9 nickase plus a reverse transcriptase plus a guide RNA with reverse transcriptase template added to its 3' or 5' end could template reverse transcription of the nicked target DNA site, and he ran all the proper controls to show that these weren't artifacts. And then he tried this in yeast cells, and it worked quite well in yeast, which was starting to get interesting. However, yeast are addicted to all sorts of DNA repair processes that are very difficult to do in humans, such as homologous recombination, which is really easy to do in yeast, but very difficult to do in humans. In fact, yeast prefer to make homologous recombination products over indels in many cases in response to double-stranded breaks, whereas the opposite is true in human cells. So we were sort of cautiously starting to get excited, but we really didn't know if it would work in mammalian cells. And so then he started trying in mammalian cells, and his first experiments were all complete failures. 0% editing, not even 0.5% editing. And Andrew very quickly figured out what changes in the system were needed to get it to work in mammalian cells. And then I remember in one Monday subgroup meeting, people walking to my office. Every Monday, everybody in the lab presents in alternate Mondays. So everyone presents every other week, what they've been up to, what problems they've encountered, what results, positive or negative, they've garnered over the past couple of weeks. And the lab helps brainstorm ideas for what to do next, how to fix problems, etc. And as we were walking in, as the students were walking into my office that Monday, one of the students looked at me as he walked by and said, "You are going to freak out," which is usually a good thing to hear before one of these meetings. And I said, "Oh, why?" And he said, "Because Andrew has data in mammalian cells." And so Andrew presented the first prime editing data in mammalian cells. And, it was very modest. It was a few percent editing. But the fact that we could now install transversions and insertions and deletions for the first time at targeted sites in mammalian cells was electrifying and just caused a sort of buzz in the group and an excitement and all sorts of brainstorming about what to do next. That was, probably unlike anything I've seen in the 22 years I've been a professor.
Sure, no, I bet it is. And in particular, I think the most remarkable thing about the prime editing design is the fact that you extended the guide RNA. And so the guide RNA not only contains the primer site for the reverse transcriptase, but it also contained the desired edit changes. And so what that meant is you didn't need to introduce a donor template that had the desired edit. And so in many ways, you kind of simplified the system as well.
Right. And one of the several things that had to work out in order for it to be successful is, so as you point out, there's a primer binding site that has to complement the nicked DNA strand so that it aligns the nicked DNA strand here. So this part is use my YouTube, use my Zoom here. So this part is the primer binding site. Here's the nicked DNA strand. And then the rest of my right hand is the reverse transcriptase template. And so this structure has to align, and reverse transcription then goes in this direction to copy the edited DNA sequence directly into the target DNA strand of the human cell genome. However, this primer binding site here, therefore, mathematically must complement the spacer part of the guide RNA. The guide RNA still has to do its main job of programming the CRISPR protein to engage the target DNA site. And so the primer binding site is complementary to the spacer, which means maybe all that a PEG RNA, a prime editing guide RNA, would end up doing is forming a sort of stem loop.
I see, yeah.
So that was, one of the risks. And indeed, we found that you have to be very careful how long that primer binding site is. You make it too long, even though normally polymerases love having long primer binding sites because it provides a bigger runway more stable runway to initiate DNA polymerization. In this case, prime editing is all about walking the line between a variety of boundary conditions and threading that needle so that you can initiate these molecular events without forming structures that would ultimately be non-productive. So the primer binding site has to be of a moderate length, long enough to support reverse transcription, but not so long that it ends up impeding the spacer. Anyhow, so all of that ended up actually working out. And after some improvements and a lot of testing in lots of different gene sites and in different cell types, we reported prime editing at the end of 2019. And it's been subsequently, and then of course, shortly thereafter, everything shut down because of the pandemic. But our lab and others have continued to use and advance prime editing. And now there have been a number of improvements, some of which have been published by other labs, others of which we hope to report this year, that following in the footsteps of base editing, that have advanced prime editing towards broader and broader applicability and greater and greater efficiency. And one thing that I think the roughly 30 or so prime editing papers or preprints that have already been published seem to point to is the DNA specificity of prime editing really does appear to be very high, higher than that of traditional CRISPR gene editing platforms, because probably in order for the prime edit to actually take place, Watson-Crick base pairing has to occur not just between the spacer and the protospacer the spacer of the guide RNA and the protospacer, the target site of the DNA, but also of the primer binding site and the nicked DNA strand, and then downstream between the 3' flap and the original DNA strand. So there's 3 different checks on DNA hybridization on sequence fidelity that each serve as an opportunity to reject an off-target sequence. And we think that's why prime editing has now been observed in multiple labs to be extremely DNA sequence specific.
That's definitely an added benefit then, right? And as well, at least in the original prime editing paper, as you said, with base editing, one of the problems is you end up with a mismatch, right, where one strand's got the edit and the other one doesn't. And so you want to kind of bias the cell to choose the strand with the desired edit. And the way that you did that was by having a secondary Cas9 complex that induced a nick, like a bit downstream. And so have you find ways, obviously the concern with that is that you've now got a nick in 2 places, which could cause a staggered double-stranded break, which is something that you would maybe kind of want to avoid. And so have you found ways of circumventing that? Would that be an issue in terms of being able to translate prime editing to the clinic?
Yeah, so if you nick that bottom strand, you do increase prime editing efficiencies, but you also increase indel levels. Indel levels are generally, with that doubly nicked version of the prime editing system, are generally higher than that of base editing, but with still favorable ratios of editing to indels. So it might be, for example, you get 30% prime editing and 8% indels or something like that. So we have improved that aspect of it in a couple of ways, which we hope to report this year. One is we have identified some of the proteins that are implicated, that are involved in the prime editing intermediate advancing and resolution, and have manipulated those proteins to favor the desired outcome. And that tends to increase prime editing efficiencies in many cases to the point that you don't need the bottom strand nick anymore to get sort of therapeutically relevant levels of prime editing, even in some therapeutically relevant cell types, like T cells or primary patient, certain kinds of primary patient cells. Also, we have developed variants of, I'd say, we've become better at how to use the prime editing system to program that second nick so that it only occurs after the first strand has been repaired, which minimizes the coincidence of having 2 nicks. And then finally, we've also gotten better at choosing where to put that second nick so that it minimizes the chance of that the 2 nicks turn into a double-stranded break. Nicks occur all over your genome quite frequently. Yeah. It's estimated that, at any moment, there might be hundreds, if not thousands, of nicks in your genome. So the trick to avoid turning nicks into double-stranded breaks if they're on opposite strands is to make sure that the nicks either never occur at the same time, or if they do occur at the same time, they're far enough away that there's plenty of Watson-Crick base pairs to hold the double helix together, and to avoid having the ends come apart into a double-stranded break. So it's a great question, and there are, I think, a number of approaches that some published, some not yet reported, that improve that aspect of prime editing.
Cool. I think one of the things that fascinates me most about just the CRISPR-Cas9 system is the number of ways that you can kind of modify it. So as you've mentioned, you can modify not only the Cas9 protein itself by mutating it, you can also modify the guide RNA as demonstrated with prime editing. And so I guess one of the major reasons why I'm interested in it is during my master's project, I was actually working on something very similar, whereby I was also trying to perform precise genome editing by having only a single-stranded break, so instead of a double-stranded break. And my approach was slightly different and involved fusing Cas9 to just a variety of different protein domains and seeing what would work. Obviously, that work I don't think would ever get published now because, you've got prime editing But it was interesting to
You should
see.
you should publish it. You should publish it. I think it's still interesting. In fact, we had a vaguely similar project where Holly Reese in my lab found that fusing a Cas9 nickase to a RAD51 protein could increase the efficiency of single-strand Nick-directed HDR in certain circumstances. But I think all of these tools and all of the insights that come from their development contribute to the field. So I think you should publish that work.
Maybe we should talk about this afterwards or something.
Encourage your PI to advance it.
But yeah, I think the one thing that I did with my project is I came up with an alternative name for it. So the name that I went for was Cas9She, which I thought was quite catchy. So that's SHEE, and it stood for safe and high efficiency editing, which is, I guess, the two problems we're trying to solve with genome editing. And then there was a kind of catchphrase that went along with it, which was Cas9She is key for genome editing. I used to put it in my slides to see if anyone got it, but yeah, I had a bit of fun with that. So going,
science needs more puns.
It's true. So going, one thing I haven't spoken about yet, which I'm quite interested in, is a big problem with genome editing, besides safety, besides efficiency, is actually being able to deliver these complexes into the side cells. And so there are also ex vivo approaches, but I think that the major challenges at the moment are more with in vivo approaches, so getting them into human patients. And so, at least from my understanding, there are two main ways in which this can be done. The first one is by using adeno-associated viruses, and the second one using lipid nanoparticles. And so I was wondering if you could talk about both of those, what you think is more promising, will it vary, which one to use depending on the genetic disease?
Right. I think just like the editing agents themselves, the delivery modalities, we will need and eventually use a whole library of different ones. There are an endless variety of lipid nanoparticles that have been shown to deliver to various organs in vivo. They can deliver messenger RNA, they can deliver DNA, they can deliver the RNP, the protein RNA complex. And some have been used with remarkable results. Verve Therapeutics, in collaboration with Beam, reported the use of LNPs to deliver RNAs encoding base editors into primates to base edit PCSK9 to cause a drop in PCSK9 protein levels that results in lowering of serum LDL cholesterol and triglyceride levels, which of course are strong correlates of cardiovascular disease. And on the AAV side, our lab reported recently the use of AAV injected into the bloodstream of a mouse with the human progeria gene to directly base edit. So the AAV encodes a base editor. The base editor corrects the mutation that causes progeria in people and in mice, and rescues quite strongly many of the phenotypic manifestations of progeria that are seen in these mice, some of which parallel the consequences of progeria in human patients. So those are examples, but I think going forward, there are now hundreds of AAV variants that researchers have reported that have all sorts of different tissue tropisms and other different properties. There are dozens, if not hundreds, of lipid nanoparticles that researchers have reported that likewise have different biodistribution properties, different half-lives, different delivery efficiencies. And so I think even just those two modalities will likely play a fairly important role going forward, and an increasingly diversified role, being able to deliver these macromolecular machines or the nucleic acids that encode them to a variety of targeted tissues. Cool.
And so the other questions that I want to do is kind of taking a bit of a sidestep is the fact that on this channel a lot I talk about targeting aging and finding ways to target aging and effectively age-associated diseases. And there's a lot of work being conducted at the moment in terms of looking at epigenetic marks and its association with aging. And so I was just wondering, what are your current opinions on the potential for precise epigenome editing by using maybe CRISPR-Cas9 approaches? For example, instead of having the Cas9 fused to a adenine-based editor, you instead have a demethylase enzyme or an acetylase enzyme or something completely different.
Right, and researchers have done exactly what you've just described, and there are multiple systems now, including one just reported or being about to be reported by Jonathan Weissman and Luke Gilbert and their co-workers. I think Jonathan's given a bunch of talks on the topic, so I suspect he wouldn't mind me talking about it. But the Weissman system is called CRISPR-off, and it uses a cutting incompetent Cas9, like we use in our base editors, a dead Cas9, fused to a variety of very carefully and cleverly chosen proteins to programmably turn off a whole region of DNA, and it does so in a heritable way, in a way that's durable, which is really quite cool. So it's not as though you necessarily have to administer for the rest of the lifetime of the organism or the patient the editing agents to get a long-lasting change in gene expression. But I generally agree that targeted epigenetic modulation is one of the great frontiers of this new kind of democratized, programmable way of looking at the genome. And in the interest of full disclosure, I have co-founded a company in this space called Chroma Medicine with Jonathan Weissman, Keith John, and Luke Gilbert. So to be transparent, I certainly believe enough in the potential of those systems to be involved in trying to translate them into patient benefit.
Cool. And you mentioned one question I was going to ask you about, which is the heritability of the fact that obviously with genome editing, it kind of makes sense, right? If you've managed to edit the genetic DNA sequence, it can get passed on through the semi-conservative replication to next cells. But epigenetic changes, as you say, it can be questionable about whether or not they would be permanent. But I think our understanding of the epigenome obviously is improving all the time. And cell states, which pretty much governs which genes are being expressed in different cell types, and cellular reprogramming, going from a differentiated cell back to a stem cell, are all things that potentially could be mediated by just making maybe a few precise epigenome marks that could propagate throughout the rest of the cell. I don't know what your thoughts are.
Right. No, I totally agree. And I think one of the really cool scientific discoveries of the past few decades, in my opinion, has been the discovery of ways that epigenetic marks are maintained over time, so that changes to the state of DNA modification or proteins that regulate gene expression are not simply modulating gene expression only at the moment that they're installed, but rather they can actually be passed down to progeny cells as well. So, I mean, in a sense, it's the core definition of epigenetics that you can regulate changes in biology, even that are heritable, that don't necessarily require changes in DNA, and that phenomenon is both now pretty well established, is highly relevant for treating potentially genetic diseases, as well as doing a lot of cool research in biology and genetics, and I think is poised to be one of the great new frontiers of the sort of programmable taking control of our genomes space.
Cool. So you kind of led on to my next question, which is, in like 10 years' time, what kind of big events or do you hope what are you excited for in the next 10 years in terms of scientific breakthroughs? That's a bit of a big question, but.
Yeah. I mean, I hope within 10 years we will see many of the technologies, if not all of the technologies we've touched on during this interview, benefit patients. And at the rate of progress, which shows no signs of slowing down and only signs of speeding up, I think it's easy to predict that. I mean, it's only been 9 years since the very first paper that reported Jennifer Doudna, Martin Janik, Emmanuel Charpentier's paper reporting the use of Cas9 for genome editing. And it's only been 5 years since our first base editing paper, and only been a year and a half since our first prime editing paper. But these tools are very, very widely used and advanced, and companies have already been founded to take all of these technologies to the clinic to benefit patients. In the case of the nucleases, as I mentioned, they're already in people. There are already humans walking this earth who are experiencing for the first time a relief of some of their symptoms of a genetic disease because they have CRISPR edited DNA inside their body. Edited ex vivo at first and then transplanted, but there are also people who are in clinical trials now who have had the first in vivo CRISPR editing take place in the body in a clinical trial that Editas has run, for example. So, I think it's easy to predict that within 10 years, there should not only be many, many more such trials, but there should be some actual drugs on the market that are benefiting patients suffering from, I think, a fairly wide range of diseases with a genetic component. I'd be surprised and disappointed if 10 years from now that weren't the case. Hopefully programmable epigenetic modulators will begin to be clinically used as well by then. So I with the caveat that I think academics end up being pre-selected to be optimists, because if you don't believe all of this stuff is going to work eventually, then what's the point? But I think 10 years is a plausible time frame to see most or all of these technologies benefit patients in a very direct way.
Cool. So that's super exciting. So thank you very much for taking the time to talk to me today. I'm very excited to hear about your work in the coming years, and I really hope that next time we'll be able to talk in person as opposed to through Zoom. But yeah, it was a pleasure to talk to you.
Likewise. I would enjoy that. I really hope you keep doing these interviews. I think you're an outstanding interviewer, and you have a really broad and sophisticated perspective on science. And I hope your readers and your following continue to appreciate the work that you're doing. So please keep up the important communication of science that you've taken up.
Thank you. So thank you for making it to the end of this interview. I hope you enjoyed it, and I hope to continue doing them in the future, as I had some fun with it. So as always, I hope you've learned something in this video. Thank you to my Patreon supporters, and thank you for listening.